Assalamu alaikum. I am Dr. Sharmin Rahman, Associate Professor, Department of Anatomy at the NMS Medical College. Today I am going to discuss about the muscle tissue. Muscle tissue is composed of cells that are specialized to shorten in length by contraction. Muscle cell is called myocyte. Myocytes are elongated in one direction and are therefore often referred to as muscle fiber. Microfilaments and associated proteins together generate the forces necessary for cellular contraction, which drives movement within certain organs and the body as a whole. There are some terminology related with muscle tissue, such as muscle cell membrane is referred to as sarcolemma. The cytoplasm of muscle tissue is all known as sarcoplasm. The smooth endoplasmic reticulum, known as sarcoplasmic reticulum, and the mitochondria of muscle tissue are known as sarcosomes. On the basis of morphological and functional characteristics, muscle tissue can be divided as skeletal muscle, cardiac muscle, and smooth muscle. Skeletal muscle is composed of bundles of very long, cylindrical, and multinucleated cells that show cross striations. Their contraction is quick, forceful, and usually under voluntary control. Cardiac muscle also has cross striations and is composed of elongated, branched, individual cells that lie parallel to each other. At sites, end-to-end contact are intercalated discs found only in cardiac muscle. Contraction of cardiac muscle is involuntary, vigorous, and rhythmic. Smooth muscle consists of collection of fusiform cells that do not show striations. Their contraction process is slow and not subject to voluntary control. Now we are going to discuss about skeletal muscle. The entire muscle is surrounded by epimysium, a dense irregular collagenous connective tissue, and perimysium, a less dense collagenous connective tissue derived from epimysium, surrounds bundles or fascicles of muscle fibers. Endomysium, composed of reticular fibers and an external lamina or basal lamina, surrounds each muscle cell. In this figure, we can see this is a muscle fiber which is surrounded by endomysium and some muscle fibers collectively known as fascicle, which is surrounded by perimysium, and some fascicles forms the whole muscle or total muscle that is surrounded by epimysium. There is another picture where we can see muscle fibers surrounded by endomysium and fascicles are surrounded by perimysium and whole muscle is surrounded by epimysium. Now microscopic structure of skeletal muscle. Under light microscope, longitudinally sectioned skeletal muscle fibers show cross striations of alternating light and dark bands. The darker bands are called A-band, that are anisotropic or birefringent in polarized light, and the lighter zone or lighter band are called I-bands, that are isotropic, do not alter polarized light. In the transmission electron microscope, each I-band is seen to bisected by a dark transverse line that is known as Z-line. The region of the myofibril between two successive Z-line discs known as a sarcomere, which is the contractile unit of skeletal muscle fiber. The I-bands consist of the portion of thin filaments, that are actin filaments, that do not overlap the thick filaments, that is myosin filaments. And the A-bands are composed mainly of thick filaments in addition to overlapping by portions of thin filaments. These are thick filaments, that is myosin, and these are thin filaments. These are thin filaments that are actin. Here we can see A-band is composed of mainly thick filaments, and peripherally they are overlapped by thin filaments, and I-band only composed of thin filaments. A-band shows the presence of a lighter zone in its center that is known as H-zone, and this H-zone is bisecting by a line that is known as M-line. So, in this figure we can see this is darker zone, that is A band, and in this darker zone in the center there is lighter zone that is known as H zone, and this H zone is bisected by a line that is known as M line, and this lighter zone is known as I band, which is bisected by a transverse line known as Z line or Z disc, and two successive Z disc area is known as sarcomere, which is the contractile unit of skeletal muscle. So we can say sarcomere is consist of one A band and on each side half I band. To provide for a uniform contraction, skeletal muscle fibers have a system of transverse or T-tubules. These are transverse or T-tubules. These finger-like invaginations of the sarcolemma form a complex network of tubules that encircles every myofibril near AI band boundaries of each sarcomere. Adjacent to opposite sides of each T-tubule are expanded terminal cisternae of the sarcoplasmic reticulum. This specialized complex, consisting of a T-tubule and usually two small cisternae of sarcoplasmic reticulum, is known as triad. This is transverse tubule or T-tubule on each side. These are terminal cisterna of sarcoplasmic reticulum. This tree is known as triad. Now cardiac muscle. These are branching fiber. They exhibit a cross striation. Each cardiac muscle cell possesses only one or two centrally located pale staining nuclei. Presence of dark staining transverse lines that cross the chains of cardiac cells at irregular intervals, known as intercalated disc. This is a cardiac muscle fiber which shows cross striation, and these are branching, and these are nucleus, and these are intercalated disc. Intercalated disc. Transverse regions of these step-like discs have many desmosomes and fascia adherens, which resemble the genula adherens between epithelial cells. Together, these two junctions serve to bind cardiac cells firmly together to prevent their pulling apart under constant contractile activity. The more longitudinal portions of each disc have multiple gap junctions, which provide ionic continuity between adjacent cells. These act as electrical synapses and allow cells of cardiac muscle to act as in a multinucleated syncytium, with contraction signals passing in a wave from cell to cell. These are gap junctions, which are present in longitudinal region, and these are desmosomes. These gap junctions helps to transfer ion one cell to another cell, and this desmosome helps to prevent a part of the cardiac muscle fibers during contraction. This is another simplified figure where we can see these gap junctions present in longitudinal section, and this in transverse section there are presence of desmosome and fascia adherens. The sarcoplasmic reticulum of cardiac muscle does not form terminal cisternae. Instead, small terminals of sarcoplasmic reticulum approximate the T tubules. These patterns resulting a dyad. Now, smooth muscle. These are elongated, tapering, and non-striated cells, each enclosed by a thin basal lamina and a fine network of reticular fibers. Each cell has a single nucleus located in the center of the cell. A rudimentary sarcoplasmic reticulum is present, but tubules are not. This table shows differences between skeletal muscle, cardiac muscle, and smooth muscle. Sarcomeres and myofibrils present in skeletal muscle and cardiac muscle and absent in smooth muscle. In the skeletal muscle, these are multinucleated and peripherally located. Cardiac muscle has one or two centrally located nucleus, and on smooth muscle, single nucleus centrally located. Sarcoplasmic reticulum are well developed with terminal cisterns. In cardiac muscles, these are poorly defined and some small terminals present, and in smooth muscle, some smooth endoplasmic reticulum present. T-tubules present in skeletal muscle, small and involved in triad formation, and in cardiac muscles also present, large and involved in dyad formation, and it is absent in smooth muscle. Cellular junctions present only in cardiac muscle, that is in intercalated disc. Contraction. Skeletal muscles are voluntary, all or none. Cardiac muscles are involuntary, rhythmic, and spontaneous. Smooth muscles are involuntary, slow, and forceful, not all or none. Calcium control. Calsequestrin in terminal cisternae. In cardiac muscle, calcium from extracellular sources and the sarcoplasmic reticulum. In the smooth muscle, calcium from extracellular sources and the sarcoplasmic or endoplasmic reticulum. Calcium binding protein troponin C present here and also in cardiac muscle, and in smooth muscle calmodulin. In skeletal muscle and smooth muscle, regeneration present, and mitosis is present in smooth muscle, and nerve fibers, somatic, motor, in cardiac muscle autonomic, and in smooth muscle also autonomic supply. Connective tissue, in skeletal muscle, presence of epimysium, perimysium, and endomysium, and in cardiac muscle, connective tissue sheaths and endomysium. In smooth muscle, connective tissue sheaths and endomysium present, and skeletal muscle is long, cylinder shaped, and many peripheral nuclei present, and cardiac muscles are branched cells, intercalated disc, and one or two nuclei, and smooth muscle cells are fusiform cells with no striation and single nucleus. That's all for today. Thank you.